Well, hi and welcome back. All right, we are going to start our next unit for our next midterm exam, which is going to focus primarily on aspects of cells. Pretty much any type of cell, whether you're looking at a simple bacterium or the multitude of cells that you might find in any multicellular organism, whether that's a tree or a blue whale. There are a lot of things about cells that they all have in common. And so that's really what this class is meant to teach you, or what are the fundamental principles that allow any living thing to function. And the thing about cells is that they are the fundamental units of life. They are the structures where all of life functions occur. Cells are the smallest units to exhibit all of the properties of life. So these are the things that are taking in energy and other resources to fuel a metabolism that then allows the cell to actually maintain a homeostatic balance and respond to stimuli. Cells can grow, cells reproduce, cells evolve. So everything that really happens in biology kind of ultimately happens at the level of cells. And so we need to spend a lot of time understanding what cells are and how they all basically work. Now, you might notice at this point, if you're looking at the screen, that we're kind of skipping ahead a chapter. We're actually going to chapter 7 after most recently covering chapter 5. And this is kind of by design on my part, because if you're going to talk about cells and you're going to talk about how cells are made, the most fundamental material used in cellular construction are these things called membranes. And so I think in order to better understand all of the remaining structures of a cell, you need to have a solid foundation and understanding of what cellular membranes really are, what they're made of, and what they're used for. And so that's why I want to actually start with this topic. And then after we cover a good understanding of membrane structure and function, we're going to go back to chapter 6, and we're actually going to understand the origins and evolution of cells a little bit, the basic varieties of cells, and then go through their fundamental structural and functional characteristics. So that is the plan for these next couple of chapters. And then after that, we're going to take a look at understanding how cells primarily take care of energy. It's kind of the main thing. And where does energy that fuels their metabolism comes from? And so we'll spend some time talking about the basics of cellular energy and even basic principles of energy that have to be met in order for cells to use energy to keep themselves alive. And then we'll talk about the way that energy is harnessed and stored and utilized. And so we'll be talking about those aspects, particularly the topics of cellular respiration and photosynthesis. But we also have to talk a little bit about one of the major uses for energy in cells, which is to provide energy for chemical reactions. And it turns out that it could take a lot more energy to make life function than it really does. And the reason we can get away with that is that we have special proteins called enzymes, which help to lower some of the energy costs necessary for metabolism to work. And so we're also going to talk about enzymes and their role in cellular metabolism. So it's all going to kind of come together over these next several chapters, and then we will get to exam number 2 after we discuss the basic principles of photosynthesis. Okay, so let's jump right in to chapter 7, membrane structure and function. Okay, well, actually, let's start with function. Okay, let's understand what are the basic roles that membranes have in cells. Because they are critical to so many basic cellular functions. First and foremost, you have what we call plasma membranes. This is the outer boundary of any cell. And so in a lot of ways, this is what physically defines the cell. It separates the cell from the external environment, as well as separating it from any nearby or adjacent cells, which may be somewhat linked to one another if you're talking about a multicellular organism, although you'll be surprised at just how much space there is even in multicellular organisms between adjacent cells. So plasma membranes define the cell. They also help the cell to interact with its environment. Part of the structure of cell membranes allows it to detect the presence of particular molecules and then respond to that. Membranes can also sometimes respond to physical aspects in the environment. To a certain extent, it can respond to forces of temperature and pressure. Even light can be influenced by membranes. Membranes are also important in helping cells communicate with one another, often by being the location for special proteins that we call receptor proteins that then bind to molecules released by neighboring cells as a form of chemical communication. And by far, one of the more significant roles of the plasma membrane for any cell is that it helps to control what can come into the cell or what can exit the cell. Because in order to have a functional metabolism, that requires a very particular concentration of various molecules, and only allowing certain substances to come in so that you don't interfere with those biochemical reactions. Also need to be able to expel materials that if they're waste products that could become toxic, you need to be able to get those out of the cell. So the membrane is the gatekeeper for what goes in and out of the cell. And now that's just the plasma membrane, which is the outer portion of any cell's membrane. Most cells, with the exception of some of the simple prokaryotic cells that we call bacteria, most cells have a whole bunch of internal membranes, and those play a very important role in compartmentalizing the cell so that different sections of the cell can specialize in particular aspects of life functions, whether it's certain areas are relegated to performing protein synthesis. Others create little compartments for intracellular digestion or breakdown of macromolecules. And in fact, many of the functionally structured elements within a cell that we call organelles, just meaning like tiny organs, many organelles are actually membranous in structure. Now, most of that information is what we'll be coming back and talking about when we return to chapter 6 in a few sessions. But the basic structure of those internal membranes is very much the same as the outer plasma membrane. So we can somewhat generalize. When we talk about cell membranes, they all follow the same basic pattern. And then there's just individual differences depending on where is that membrane located in the cell and what is its overall job or purpose. So now we do need to talk about, well, how are these membranes put together? Because the capacity of a membrane to provide any of these functions is made possible due to the unique structure of cellular membranes. So let's start talking about that. So all cellular membranes are based on a structure we call a phospholipid bilayer. So this was one of the biomolecules that we looked at in chapter 5 a few weeks ago. So remember, phospholipids are these really weird molecules that are what we call amphipathic. Okay? Amphipathic just means that they have hydrophobic and hydrophilic regions. They have areas that like to be in contact and surrounded by water molecules, and they have other areas that don't want to be around water molecules. All right. Another way to say that, though, and why are they hydrophilic or hydrophobic? Well, it's because the hydrophilic regions have polar regions. Remember our previous discussion, the phosphate group that is attached to the glycerol head of a phospholipid molecule, the phosphate group is electrically charged, has an extra electron. And then there are additional functional groups that are often added onto that phosphate group. And all of that is very polar. And so it's very easy for charged regions of water molecules to surround that portion, creating like a little miniature hydration shell. The nonpolar portions, of course, are the fatty acids. Right? The fatty acids are really what allow for phospholipids to produce the bilayer, the double layer, because the hydrophobic areas want to cluster together. And this allows the basic structure to form. All the hydrophobic tails, the fatty acids, all align towards one another, creating a relatively water-free zone right here, and surrounded by the hydrophilic heads on opposite sides. So this lets all the molecules kind of cluster together, but they're not attached to each other. There are no permanent bonds between adjacent phospholipid molecules, not in the hydrophobic tail regions, and not even among the hydrophilic heads. They're just there by mutual consent, so to speak. All of these molecules, their fatty acid tails just want to huddle together, and they are able to remain in a stable arrangement, but they're still in kind of a fluid state, meaning that there's a great deal of flexibility among these molecules. So I'm going to take a quick pause here, and I will be right back. I want to show you something that maybe will help illustrate this a little bit better. All right. So here is kind of a basic model of a phospholipid bilayer. So kind of what we have here, and it's getting a little old and kind of falling apart a little bit, but we can see each phospholipid molecule is represented by these white balls, which indicate the hydrophilic heads, and then these little red pieces that are the fatty acid tails, the hydrophobic tails. All right, and so the way this model shows is that I can slosh and I can move this around, and you can see the molecules all kind of shake around, but they remain in a stable position relative to one another, almost no matter how I rotate the model around. Things are not fixed into place, but we still kind of get that double layer. And it looks like my lower layer of phospholipids, all their tails aren't quite pointing perfectly up inwards, but you can see that they're largely avoiding poking out past where the heads are. Mm-hmm. So this is the fluid nature of membranes. It holds a pretty consistent shape, but the molecules can move and flex. And this is a very important characteristic of cellular membranes, is it gives the cells a certain amount of flexibility. All right, let me go back to our slideshow here. All right. So one thing, though, that can be a little bit different depending on the species that the cell is a part of, is you may have slight differences in the types of phospholipid molecules that are used to make the membrane. And in particular, if you have differences in the degree of saturation of those fatty acids. Remember that concept from chapter 5. There were saturated and unsaturated fatty acids. And it was the unsaturated fatty acids that tended to have a little bend in the hydrocarbon chain, kind of creating a little flex or a bend. Turns out that really affects the membrane structure, which in turn actually can affect the overall permeability of a membrane. Now, all phospholipid bilayers are selectively permeable. And that simply means that they will allow certain molecules can pass from one side of the membrane through the hydrophobic region across to the other side of the membrane, but other molecules don't make it through that barrier. And so this is important for cells because, again, cells have to be able to control what can come into or what can exit the cell. Phospholipid molecules are fundamentally selectively permeable, and we'll build on that. But as I said, part of this depends a lot on just how saturated the fatty acids are that make up your phospholipids. Because if you have unsaturated fatty acids that have extra, a sharp bend in the fatty acid chain, well, as those fatty acids get next to one another, those bent portions kind of push against other molecules that maybe are more saturated. And this creates more gaps in spacing, and it creates an even looser membrane structure, so that this is going to be an even more permeable kind of a situation, because there's more space for slightly larger molecules to maybe squeeze their way past all of these phospholipids and go from one side of the membrane to another. On the other hand, if you use highly saturated hydrocarbon tails, then all of those tails are all running parallel to one another, and they can be packed in much more tightly, creating a more kind of thick or viscous membrane, which becomes less permeable because now everything is so crowded in. Even small molecules might have a hard way of pushing and shoving their way around these individual phospholipids. And some studies have shown that some plants actually vary the viscosity of their membranes on a seasonal basis, so that under cold seasons, they might actually become a little more viscous in their membranes, but during warm seasons, they open things up a little bit. It's just kind of interesting the way that the metabolism of the cells themselves can be adjusted to produce different types of phospholipid molecules that are then used to construct these various membranes. Okay. Now, another way of kind of splitting the difference between these two kinds of models, where you have either a very fluid membrane or very viscous membrane, can use other lipid molecules can also be added to the phospholipid bilayer. And this is what tends to happen in animal cell membranes, where we mostly have fairly saturated fatty acids in our phospholipids, but we can still have a little more permeability by adding in cholesterol molecules that will have something like a slightly polar hydroxyl or other functional group that helps it to maintain the alignment. But most of the ring structure of a cholesterol molecule is another lipid. It's hydrophobic, and so it'll nestle in here. This is actually what gives a reasonable amount of stability and flexibility to animal cell membranes. But let's dive in a little bit deeper though. Let's talk about just, again, what do I mean by selectively permeable? Well, it sort of works like this. Permeability is really a relative property, in terms of what kind of molecules are attempting to go across the membrane. Right? So most phospholipid bilayers, especially the outer plasma membrane, tends to be permeable to either small and non-polar molecules like oxygen gas and carbon dioxide gas. Right? In both of these cases, the sharing of the electrons in the covalent bonds that are holding these molecules together is a fairly equal sharing, because, with oxygen, two oxygen atoms have equal electronegativity, so they equally share the electrons. And even between carbon and oxygen, they're close enough to one another on the periodic table. And through the double bonds that hold the oxygen to the carbon in the middle, that creates another nonpolar molecule. But they're also small, 2 or 3 atoms, that are among the smaller atoms on the periodic table, because they're near the top of the, in really the kind of the second period, that second row of the table. So they're not very big atoms to begin with, and there aren't very many of them. So the molecular weight is really low. They're tiny. And so they're small enough, way smaller than the collection of hydrocarbon tails and the glycerol heads and everything like that. So they can often kind of squeak their way through the nonpolar regions and simply move across the membrane, primarily being driven by the process of diffusion and always moving in accordance to a concentration gradient. So that seems to work. You can also have slightly larger nonpolar molecules, like we showed you. Cholesterol molecules can actually be used to help shore up the structure of the phospholipid bilayer, but other similar molecules, other steroids, can actually very easily diffuse, because even though they might have a little bit of a repulsion from the hydrophilic heads, for the most part, lipids can mix in with other lipids. And so this layer is not a barrier to steroid molecules, and they can freely diffuse in and out of cells. All right. What's less permeable might be other small particles, small polar molecules, and particularly charged particles like ionic compounds. So water molecules, which really aren't any bigger than oxygen or carbon dioxide molecules, but water is polar. And so those charged areas on the water molecules tend to get repelled by the charged regions of the hydrophilic heads. And so water molecules have a tendency to kind of be deflected away from the membrane. They might collide into it, but tend to bounce right off. Same thing with ionic particles, particularly sodium and chlorine that, are dissolved salts, have a really difficult time getting through these regions, right? Even large proteins, you can see here, which are really large molecules, have a hard time pushing enough phospholipids away to be able to cross from one side to another. And so phospholipid bilayers are just excellent materials for making a boundary that is going to separate an external environment from an internal environment. And that internal environment needs to have a unique chemistry that favors all of the biochemical reactions of a cell's metabolism. So phospholipids are definitely the way to go. The only thing, though, is that we still might need some of these substances, like these large polar molecules, and even these smaller ionic compounds can be useful to go from one side of the membrane to another, to come in or out. So how are they getting it? I mean, like water. Shouldn't we need water? Haven't we all learned that water is essential to life? And in fact, most of the mass of any living organism is actually water by weight. How does the water get in? Because that water is not just found outside the cells, it's found inside all of the cells. But how does water get in and out? Well, the answer to that question, okay, is proteins. All right? So in addition to the phospholipids, all cellular membranes contain a variety of larger proteins that are somewhat embedded into the phospholipid bilayer itself. All right? Most of these are referred to as integral proteins or integral proteins. They're integrated into the phospholipid bilayer because as part of their protein structure, the amino acid structure, there are a lot of amino acids with hydrophobic or lipid-based side groups. Remember the variable R groups of any amino acid? And you looked at and you saw that there were a number that were categorized as nonpolar because they're mostly hydrocarbon type side groups. So those hydrophobic areas can cluster together, and if they're concentrated in one part of the protein, often in the center of the protein's overall structure, then that corresponds to the hydrophobic center of phospholipid bilayers. But then you also have hydrophilic regions of the protein structure. So those amino acids on the outer parts of the structure can then extend beyond the protein surface, creating what's also referred to as a transmembrane protein. That is a protein that spans the phospholipid bilayer and sticks out on either side. All right. But just like the phospholipids themselves, these integral proteins are not bound or fixed to any other molecules. And so they're free to actually move amongst the phospholipid molecules, because again, it's a fluid environment and they can kind of shift and move positions, possibly, depending on their connections to other proteins, either inside or outside the cell. But some of them are completely mobile elements. They're kind of like, a lot of books describe them as icebergs that are drifting in a sea of phospholipids. So, you'll see these big structures. And I've got some illustrations in the next slide that will allow you to visualize this maybe a little bit better. Right. Now, in addition to the integral proteins, there are also special what we call peripheral proteins. And those are usually just attached onto one side of the membrane. Some are only attached to the inner membrane surface. So they're connected to or clustered against the phospholipids on the cytoplasm side of the membrane. Other proteins have actually been exported from the cell, but then they cling to the outer surface or the extracellular side of the phospholipid bilayer and hang out there. And usually they are somehow linked to an integral protein that is embedded within the membrane itself. So the term that we use to describe the overall structure of cellular membranes is that it is a fluid mosaic model. Again, meaning fluid means nothing is permanently connected to anything else. There's no solid structure. It's all flexible and things are all move around. And then it's a mosaic in the sense that there are periodic piercings of different types of proteins scattered all around the membrane region. Kind of like mosaic art. We're using different bits of stone or glass or ceramic that are embedded within a mortar matrix. That's like, the mortar is like the phospholipids, and then each protein is an individual stone or mosaic piece, tile piece or something. That's kind of the illusion we're getting into. Now, the proteins are there actually to do a lot of different things. And I'm going to give you just a brief list of them in just a few minutes here. But one of the more important functions of proteins is that proteins are really important in aiding in the transport of substances into or out of cells. And we'll be actually spending a lot of time just talking about that concept of transport across membranes. And we'll be talking about the role of some of those proteins. But first, let's take a look at this. Here's just a textbook illustration of a hypothetical plasma membrane. So you can see the phospholipid bilayer. And looking closely at this illustration, you can see tiny little cholesterol molecules in here. So this must be some type of animal cell protein. You can see those there. And then here are the embedded proteins. So many of these are integral proteins that you can see bridging or spanning over the phospholipid bilayer itself. And then you also have peripheral proteins that are clinging to one side of the membrane or another side of the membrane. And as we'll be getting into maybe a little bit more when we go back to chapter 6, we're also going to talk about a variety of just protein fibers or filaments that are produced by cells and then are used either internal to the cell to make up something called a cytoskeleton, essentially a structural framework that is present in all cells. Other proteins are produced and assembled to create long extracellular fibers that may be used in the case of an animal. These may actually be used to link cells to one another to form a larger tissue. So this is, if you could just magnify a plasma membrane enough, this might be something of what it would look like. And again, all of these proteins in here, many of them, if they're not connected to any of the external or internal fibers, can shift and move position within the membrane itself. And so they are, again, very fluid in nature. So let me just wrap this up and let me briefly talk about many of the different jobs that these proteins that are a part of the membrane structure, what all can they do? What are the different functions that they have? So as we'll be elaborating a lot more in the next video installment, there is transport. They are there to aid in the movement of certain substances that cannot easily pass the phospholipid portion of the membrane, but proteins can produce various channels or passageways that allow things to move from one side of the membrane to another. And this can be done either passively through mechanisms of diffusion, or that they can be transported very actively if the cell is willing to give up a little bit of energy to force the movement of molecules from one side of the membrane to another. So we're going to spend a lot more time about this function in the next video installment. But on top of that, these embedded proteins can do many other roles. One of them is some of these proteins are actually enzymes. Enzymes are the proteins that help to facilitate chemical reactions. They are able to take molecules and that are brought in as a reactant, or really what we call a substrate, is grabbed by the enzyme, and then that substrate molecule is chemically transformed into a product, and then the products can be released. Here we have a pair of enzymes that are kind of working together in a related fashion to take this, this starting molecule is captured by this enzyme, and then it is transformed into this blue particle right here, and then this can be grabbed by the next enzyme right next door and turned into this green particle right here. And this is often the way that these types of enzymes are arranged, is that since they have a related function, maybe they're part of a larger, what we call a biochemical pathway of taking certain raw ingredients and ultimately making them into a final product through a series of steps. Well, it makes sense that, well, if you have step 1, step 2, step 3, each controlled by an enzyme, you might as well link the enzymes together, 1 next to 2, next to 3. Putting them into a membrane is just going to make the process work more efficiently. So we have enzyme activity. Some of these transmembrane proteins act as what we call receptors. And they have the ability to bind to molecules that have been synthesized by other cells and then released into the extracellular environment. And these molecules drift and diffuse until they make contact, and the molecular structure of the signal molecule corresponds to the structure of the receptor. And then this leads to kind of the relay of, or the activation of, other proteins and other molecules inside the cell that are responding to the presence of this molecule. So this is how things like smell work. You know, odor molecules are like signal molecules. They bind to a receptor on our olfactory receptors in our nasal cavities. Okay, olfactory is the fancy word for smelling. So when that happens, when a scent molecule, maybe it's the scent of freshly baked bread, isn't that a wonderful smell? Most people, when they smell bread baking, they just can't help but go, good. Okay, so maybe it's a scent from some baking bread molecule, lands in our nose, binds to a receptor. This then, the signal is then translated into creating a nerve impulse that travels from our nasal lining to a region of our brain where we process that and recognize it as bread, and then whatever response we're going to have to that. Right. You're going to learn a lot about signal transduction as one of the final topics that we cover in the course this semester. So you're going to learn more about the mechanism that happens here with one of these proteins. All right. But in a related fashion, this similar mechanism can be used to help cells recognize one another. So this is really important for our immune system. How do our cells of our immune system know what is foreign and needs to be attacked and destroyed versus what is part of your own body? Well, your cells produce special proteins that have a particular arrangement of often carbohydrate chains attached to the surface, and those chains have a very particular geometry or shape associated with them. Your immune system learns to recognize those shapes as friendly. And anything that doesn't have those particular shapes is treated as an enemy. So that's a way that cells can recognize one another. Now, if you're single-celled organisms, this can be a great way of two individual cells recognizing that they are the same species and are compatible to engage in some kind of reproductive activity with one another. So cell-to-cell recognition is kind of similar to cell signaling. It's just that rather than having a separate signaling molecule, instead the two cells use their proteins to kind of see if they match each other's shapes and therefore know that they're compatible cells with each other. Another really important function is for multicellular organisms where the cells have to be very closely bound and attached to one another. Then the proteins are often used to link adjacent cells to one another. Again, having corresponding shapes on each protein that then bind and hold them together, usually not just in one spot, but in dozens and even thousands and thousands of locations along the shared membrane surfaces. So something like that. So linking cells to one another is a big role. And then also there are other tissues in organisms or in other arrangements where individual cells are not directly connected to one another, but they are attached to extracellular fibers, creating a matrix that one cell can be bound to a fiber and then another cell can be linked to the same fiber some distance away. And so we can kind of spread out the cells a little bit, but hold them all together using kind of an elaborate web of protein fibers. And then this also helps some cells maintain a very stable shape and structure because these proteins can be bound to internal fibers that are cross-linked in something we call a cytoskeleton. And you guys are going to learn more about that also when we go back and we talk about cell structure and particularly the composition and the arrangement of the cytoplasm of cells. So that's just kind of a quick preview. And a lot of these topics are going to be elaborated on in other areas of the course. So I think that is a good place to stop this installment. So where we're going to go next, we're going to come back and we're going to start talking about the whole notion of transport and how do things move in and out of cells. And we'll learn that in many cases, these embedded proteins are very important, but it turns out that they're not the only means by which substances get transported across membranes. So I hope you will join me for the next installment. Thank you for watching this one. You guys have yourselves a really great day.